Hello, in this video we will look at the ketogenesis, physiology, and end of the video we will learn about the biochemistry. So what is the ketogenesis? In this video we will learn about the ketogenesis, the process and their physiology and what is the function of the ketones. So let's begin to understand the process in which ketones produces, which is keto sugar or glyceraldehyde. So the keto sugar is I will mention basically is the functional group is the for keto and aldo. So the aldo sugar is like a glucose. So that is a glyceraldehyde, and the keto is the carbon containing double bond oxygen. So the CO bond, and in this way here is the regular sugar is basically is the also the aldehyde, but this is the aldo sugar is the lighter molecular formula, the molecular weight. So the ketogenesis occur in specific condition like exercise, diabetic person, the diabetes mellitus type 1 and 2, due to the insulin insensitivity and the fasted state. So let's begin to understand what is the type 2 diabetes. In this way, in the blood sugar, when rise, required insulin will bind with the receptor of the insulin receptor with the cell in the tissue, will lead to GLUT vesicle to transport into the plasma membrane for facilitate diffusion of glucose. But the resistance due to the receptor damage will lead to insulin resistance, will not allow to lead to hyperglycemia, not ISO. So increase the rise of the glucose level in the blood circulatory system will lead to excretion of glucose is known as glucoseuria. And after this glucoseuria, the hypoglycemia will occur, and in this way the hypoglycemia will lead to again positive feedback to degrade more muscle and the liver, the glycogen degradation, the glycogenolysis will trigger by this mechanism. But this is the fatty acid, adipose tissue, which contain triacylglycerol, and lipolysis will be occur. Due to the hormone-sensitive lipase enzyme will convert the triacylglycerol into the glycerol and the free fatty acid. This is known as the lipolysis because the lipid will be moved into the blood circulatory system. The triacylglycerol will increase, low density lipoprotein will increase, means the more lipids contain the lipoprotein, and the fatty acid will also increase. After this increasing of the low density lipoprotein will move toward the liver, the hepatic circulation. And in this way, the low density lipoprotein, basically this is the mitochondria in the liver, contain mitochondria in the hepatocyte. So in this way, here is you can see this is the cholesterol containing low density lipoprotein and free fatty acid when enter into the liver, and the PPAR alpha will trigger the beta oxidation, remember. So increasing of the beta oxidation due to the PPAR alpha. We will discuss the PPAR alpha in the next video with the detail. So what that is the PPAR? So the PPAR is the many function, but we will learn. So here is a fatty acyl CoA formation in the mitochondria will lead to acetyl CoA formation in the Krebs cycle. For the Krebs cycle, will produce more ATP, NADH and FADH2 to produce more ATP in the electron transport chain. While this fatty acyl CoA will go into the ketone bodies, production is known as the ketosis, the ketogenesis in the liver. So the ketones body can be a beta hydroxybutyrate is the major, acetone, acetoacetate, and glyceraldehyde and acetoacid. Acetoacid is come from the amino acid, remember. After this ketones production, here you can see this is the PPAR alpha also triggered by this mechanism due to the lipolysis, and will increase the FGF, fibroblast growth factor 21, will trigger the growth hormone signal decrease, so the growth hormone increase, inhibit the growth hormone signal decreasing, and the longevity and the torpor and gluconeogenesis will be triggered by this. But on the other hand, here is the ketones will enter into the blood circulatory system. So the pancreas will trigger by the ketones more production. The glucagon will trigger the muscle and liver to glycogenolysis, and insulin will decrease. So in this way, the glycogenolysis due to the fasting state or diabetic person. So this is the cell. These ketones can be directly diffused into the cell, and in this way the brain and muscle consume energy from the ketones, and ketones use efficiently than the glucose. So that is why during the fasted state there is a beneficial effect for the brain and muscle. So here is the muscle breakdown after late ketogenesis process. During the late fasting state will produce amino acid into the blood circulatory system, and this amino acid under the ketogenesis to produce aceto acid and keto acid. So now we will discuss about the biochemistry of ketogenesis. That is important. And let's begin to understand. Remember, the degradation of the adipose tissue is known as the lipolysis. The naturally, and due to the fasted state, we stimulate, or the diabetic person can be stimulated by the low glucose level, and in this way here you can see this, the adipose tissue will degrade to through a hormone sensitive lipase enzyme to produce a beta oxidation of fats, because the different hormones involved for the lipolysis. So that is why the hormone sensitive lipase enzyme used, and the beta oxidation of fat occur in the liver. So let's begin to understand the beta oxidation. So the CoA will be added in the fatty acid to form a fatty acyl CoA. After this fatty acyl CoA, the chemical reaction will occur to produce the 2-acetyl CoA. A lot of acetyl CoA, but we are taking here 2. So the forward reaction can be the CoA will be removed, while the reverse reaction can thiolase enzyme used to produce again this acetyl CoA. While the acetoacetyl CoA will be produced when the remove of one CoA to join these two molecules. After the joining to two molecule will remove the CoA, and in this way the CoA basically is the one hydrogen will be removed. So the dehydrogenation will be occur, the dehydrogenase enzyme. While here is the HMG CoA synthase can reverse reaction to produce again acetoacetyl CoA for increase the fatty acid, and in this way the HMG CoA reductase can be inhibit this reaction. So remember, the hyperlipidemia, the hypercholesteremia can be inhibited by a HMG CoA reductase drug. So in this way here you can see the beta basically acetyl CoA under the acetyl CoA addition will remove one hydrogen with the CoA, and in this way the production is the molecule is the beta hydroxy beta methyl glutaraldehyde CoA HMG CoA production. But the HMG CoA lyase enzyme, which that is can be reverse reaction, but the acetyl CoA removing to produce the acetoacetate, and this can be reversible reaction under the HMG CoA lyase enzyme, and again the HMG CoA synthase enzyme to produce acetoacetyl CoA can be reversed, but the HMG CoA reductase can be inhibited. So acetoacetate formation, this is the ketone, will under the NADH used to the hydrogenation to produce D-beta hydroxybutyrate dehydrogenase enzyme to produce D-beta hydroxybutyrate, as well as when removed the decarboxylation will lead to produce acetone. This is the ketones body, and the major is the beta hydroxybutyrate as well as acetone will enter into the blood and the blood ketones, and when rise these ketones body will lead to ketoacidosis. You can watch my video to understand about the ketoacidosis. You can search in the search bar of the YouTube channel, YouTube search bar. So this ketone will enter into the liver to blood and lead to ketoacidosis or the hyperketonuria. The and urine more production of ketones. So in this way, the exercise or the diabetic person or fasted state will stimulate